Dear viewers, this video is part 3 of the cancer, that is Neoplasia series in the Medicine for Common Man series. Now, regarding the topic hematogenous spread through bloodstream via the draining veins. Hematogenous spread is typical of sarcomas but is also seen with carcinoma. In general, histologic evidence of penetration of small vessels at the site of the primary neoplasm is an ominous feature associated with hematogenous metastasis. Several factors influence the patterns of vascular metastasis. With venous invasion, the blood-borne tumor cells often come to rest in the first capillary bed they encounter. Understandably, the liver and the lungs are most frequently involved by hematogenous dissemination because all portal area of drainage flows to the liver and all venous blood from the body flows to the lungs. Cancers arising in close proximity to the vertebral column often embolize through the paravertebral plexus of veins. This pathway produces frequent vertebral metastasis from carcinomas of the thyroid and prostate. Note: embolism is when a blood clot or a collection of cancer cells break loose and travels through the bloodstream. Nonetheless, many observations suggest that the location of the primary tumor and its natural pathways of venous drainage do not wholly explain the observed patterns of metastatic spread, which are often cancer specific. Certain cancers have a curious propensity for growth within large veins. Renal cell carcinoma often invades the branches of the renal vein and then the renal vein itself, growing in a snake-like fashion up the inferior vena cava until it sometimes reaches the right side of the heart. Similarly, hepatocellular carcinomas often penetrate portal and hepatic branches and then grow into the main venous channels. Remarkably, such intravenous growth may not be accompanied by widespread metastasis. Dear readers, by the way, this educational video is made possible by Prescribon, the makers of e-prescription software for digital prescriptions, available freely for doctors on the website www.prescribon.com. Doctors who are interested in this e-prescription can freely avail of its vast collection of more than 100,000 e-prescriptions. Benign versus malignant tumors overview. Benign and malignant tumors can be distinguished from one another based on the degree of differentiation, local invasiveness, and distant spread. Benign tumors resemble the tissue of origin and are well differentiated. Malignant tumors are less well differentiated or completely undifferentiated, that is, anaplastic. Benign tumors are more likely to retain functions of their cells of origin, whereas malignant tumors sometimes acquire unexpected functions due to derangements in differentiation. Benign tumors are slow growing, while malignant tumors generally grow faster. Benign tumors are circumscribed and have a capsule. Malignant tumors are poorly circumscribed and invade surrounding normal tissues. Benign tumors remain localized at the site of origin. Malignant tumors metastasize to distant sites. Carcinomas tend to spread via lymphatics, whereas sarcomas prefer the hematogenous route. Acquired predisposing conditions. Acquired conditions that predispose to cancer can be divided into 1. Chronic inflammatory disorders, 2. Precursor lesions, and 3. Immunodeficiency states. Chronic inflammatory disorders and precursor lesions span a diverse diverse set of conditions that are all associated with increased cellular replication, which appears to create a fertile soil for the development of malignant tumors. Indeed, repeated rounds of cell division may be required for neoplastic transformation, as proliferating cells are most at risk for somatic mutations that lead to carcinogenesis. Immunodeficiency states predisposed to virus induced cancers. Chronic inflammation. Cancer risk is increased in individuals affected by a wide variety of chronic inflammatory disease, both infectious and non infectious. Tumors arising in the context of chronic inflammation are mostly carcinomas, but also include mesothelioma and several kinds of lymphoma. As with any cause of tissue injury, these disorders are accompanied by a compensatory proliferation of cells that serves to repair the damage. Whatever the precise mechanism, the link between chronic inflammation and cancer has practical implications. For instance, diagnosis and effective treatment of Helicobacter pylori gastritis with antibiotics can quell a chronic inflammatory condition that might otherwise lead to the development of gastric cancer. Precursor lesions. Precursor lesions are defined by localized morphologic changes that identify a field of epithelium that is at increased risk for malignant transformation. These changes may take the form of hyperplasia, that is, increase in the reproduction rate of its cell, metaplasia, conversion of one adult tissue type into another, or dysplasia, that is, abnormal cells within a tissue. Precursor lesions consisting of hyperplasias often stem from chronic exposure to trophic factors. One of the most common precursors of this type is endometrial hyperplasia, which is caused by sustained estrogenic stimulation of the endometrium. A classic lesion of this type is the colonic villus adenoma, which progresses to cancer in about 50% of cases if left untreated. Most benign tumors transform rarely, example uterine leiomyomas, pleomorphic adenomas, and others not at all, example lipoma. Why most benign tumors have a negligible risk of malignant transformation is an unsettled question. One possibility is that benign tumors at high risk for malignant transformation possess the cancer enabling property of genomic instability discussed later, whereas other benign tumors do not. Immunodeficiency and cancer. Patients who are immunodeficient, particularly those with deficits in T cell immunity, are at increased risk for cancer, especially types caused by oncogenic viruses, presumably because these individuals have a higher than normal incidence of chronic infection with viruses. These viruses associated tumors include lymphomas, certain carcinomas, and some sarcomas and sarcoma-like proliferations. Epidemiology of cancer. Overview. The incidence of cancer varies with geography, age, race, and genetic background. Cancers are most common in adults older than 55 years of age, but occur in adults at all ages and in children and infants. The geographic variation is thought to mainly stem from different environmental exposures. Important environmental exposures implicated in carcinogenesis include infectious agents, smoking, alcohol, diet, obesity, reproductive history, and exposure to environmental carcinogens. The risk of cancer is increased by reparative proliferations caused by chronic inflammation or tissue injury, certain forms of hyperplasia, and immunodeficiency. Interactions between environmental factors and genetic factors may be important determinants of cancer risk. Molecular basis of cancer. Role of genetic and epigenetic alterations. 1. Evidence for the genetic origins of cancer has been building up for decades. Non-lethal genetic damage lies at the heart of carcinogenesis. The initial damage or mutation may be caused by environmental exposures, may be inherited in the germline, or may be spontaneous and random, falling into the category of bad luck. The term environmental refers to any acquired mutation caused by exogenous agents such as viruses or environmental chemicals or by endogenous products of cellular metabolism that have the potential to damage DNA such as reactive oxygen species or alter gene expression through epigenetic mechanisms, example, so-called oncometabolites. A tumor is formed by the clonal expansion of a single precursor cell that has incurred genetic damage. That is, tumors are clonal. Alterations in DNA are heritable, being passed to daughter cells, and thus all cells within an individual tumor share the same set of mutations that were present at the moment of transformation. There are 4 classes of genes. 1. Growth promoting proto-oncogenes. 2. Growth inhibiting tumor suppressor genes. 3. Genes that regulate programmed cell death, that is technically known as apoptosis. And 4. Genes that are responsible for DNA repair. These are the principal targets of cancer-causing mutations. Carcinogenesis results from the accumulation of complementary mutations in a stepwise fashion over time. Figure 1.4 shows cancer development through stepwise acquisition of complementary mutations. Initially, carcinogens induce mutations. Then, over time, they acquire favorable additional mutations to become a cancer-causing cell. Later, again over a period of time, they acquire additional mutations for invasiveness and metastasis. See the figure 1.4 again. Malignant neoplasms have several phenotypic attributes referred to as cancer hallmarks, such as excessive growth, local invasiveness, and the ability to form distant metastases, which stem from genomic alterations that change the expression and function of key genes and thereby impart a malignant phenotype, that is, observable behavior. Mutations that contribute to the acquisition of cancer hallmarks are referred to as driver mutations. The first driver mutation that starts a cell on the path to malignancy is the initiating mutation, which is typically maintained in all the cells of the subsequent cancer. However, because no single mutation appears to be fully transforming, development of a cancer requires that the initiated cell acquire a number of additional driver mutations, each of which also contributes to the development of the cancer. The time over which this occurs is unknown in most cancers, but appears to be lengthy. Even in aggressive cancers that clinically seem to appear out of the blue, such as childhood acute lymphoblastic leukemia, cells bearing initiating mutations may be found in blood samples taken as long as a decade before diagnosis. The persistence of initiated cells during this long preclinical prodrome is consistent with the idea that cancers arise from cells with stem cell-like properties, so-called cancer stem cells, that have a capacity for self-renewal and long-term persistence. Loss of function mutations in genes that maintain genomic integrity appear to be a common early step on the road to malignancy, particularly in solid tumors. Note, Loss of function mutation, they are also called amorphic mutation or null mutation. They are a type of mutation in which the altered gene product lacks the molecular function of the wild type that is naturally occurring gene. Mutations that lead to genomic instability not only increase the likelihood of acquiring driver mutations, but also greatly increase the frequency of mutations that have no phenotypic consequence, so called passenger mutations, which are much more common than driver mutations. As a result, by the time a cell acquires all of the driver mutations that are needed for malignant behavior, it may bear hundreds or even thousands of passenger mutations. Note, driver mutations versus passenger mutations. Driver mutations, mutations conferring a fitness advantage to somatic cells in their microenvironment, thereby driving the cell lineage to cancer. Conversely, passenger, also termed hitchhiker's mutations, are defined as mutations providing no such proliferative benefit. Mutations in many other genes contribute to tumorigenesis by interfering with host immune responses or altering interactions with the stroma or by other mechanisms. Once established, tumors evolve genetically during their outgrowth and progression under the pressure of Darwinian selection, that is, survival of the fittest. Early on, all the cells in a tumor are genetically identical, being the progeny of a single founding transformed cell. However, by the time a tumor mass comes to clinical attention, generally when it attains a mass of about 1 gram or about 10 power 9 cells, it has gone through a minimum of 30 cell doublings. This number is actually a substantial underestimation because a fraction of cells in all tumors dies by apoptosis during preclinical stages of tumor development. During the expansion process, individual tumor cells acquire additional mutations at random. As a result of this tumor evolution, even though cancers are clonal in origin, by the time they become clinically evident, their constituent cells are often extremely heterogeneous genetically. See Figure 1.4. These diverse tumor subclones compete for access to nutrients and microenvironmental niches, and those that are most fit win this Darwinian struggle and come to dominate the tumor mass. This pernicious tendency of tumors to become more aggressive over time is referred to as tumor progression. In the case of species, that is, evolution of species, this genetic divergence occurs over a period of many millennia, that is, thousands of years, whereas in tumors, subclones may arise and diverge on a time scale of years, months, or even weeks. Selection of the fittest cells can explain not only the natural history of cancer, but also changes in tumor behavior following therapy. One of the most profound selective pressures that cancer cells face is effective therapy. Tumors that recur after therapy are almost always found to be resistant if the same treatment is given again, presumably because therapy selects for subclones that by chance have a genotype that allows them to survive. In addition to DNA mutations, epigenetic aberrations also contribute to the malignant properties of cancer cells. Epigenetic modifications include 1. DNA methylation, which tends to silence gene expression, and 2. Modifications of histones, the proteins that package DNA into chromatin, depending on their nature, may either enhance or dampen gene expression. Cellular and molecular hallmarks of cancer. Over the past several decades, hundreds of genes that are mutated in cancer have been discovered. It appears that all cancers display 8 fundamental changes in cell physiology, which are considered the hallmarks of cancer. These changes are the following: 1. Self sufficiency in growth signals. Tumors have the capacity to proliferate without external stimuli, usually as a consequence of oncogene activation. 2. Insensitivity to growth inhibitory signals. Tumors may not respond to molecules that inhibit the proliferation of normal cells, usually because of inactivation of tumor suppressor genes that encode components of growth inhibitory pathways. 3. Altered cellular metabolism. Tumor cells undergo a metabolic switch to aerobic glycolysis called the Warburg effect, which enables the synthesis of the macromolecules and organelles that are needed for rapid cell growth. 4. Evasion of apoptosis. Tumors are resistant to programmed cell death. 5. Limitless replicative potential, that is, immortality. Tumors have unrestricted proliferative capacity, a stem cell like property that permits tumor cells to avoid cellular resistance, that is, cellular senescence, and mitotic catastrophe. 6. Sustained angiogenesis. Tumor cells, like normal cells, are not able to grow without a vascular supply to bring nutrients and oxygen and remove waste products. Hence, tumors must induce angiogenesis. 7. Ability to invade and metastasize. Tumor metastases are the cause of the vast majority of cancer deaths and arise from the interplay of processes that are intrinsic to tumor cells and signals that are initiated by the tissue environment. 8. Ability to evade the host immune response. The cells of the innate and adaptive immune system can recognize and eliminate cells displaying abnormal antigens. Example, a mutated oncoprotein. Cancer cells exhibit a number of alterations that allow them to evade the host immune response. These are considered enabling characteristics because they promote cellular transformation and subsequent tumor progression. Dear viewers, this is the end of part 3 on cancer, that is neoplasia, in the Medicine for Common Man series. Thank you for watching the video.